Awesome. Yeah, thanks so much for having me, you guys. And thanks so much to Flora and Andre for setting this up. I met Flora when we were on our way to ABRC in the airplane in Mobile, Alabama. So that was a really fun connection to make. Um, but yeah, so I'm going to talk to you today about entomopathogenic fungi. Um, if it's tough to remember that word, I usually tell people to break it down in by thinking of it as entomo, like entomology, and then it's a pathogen. So entomopathogenic fungi, which are essentially our fungi that naturally infect and kill insects and arthropods. Um, so a little background on me. I originally wanted to study fungi in graduate school, and I had been working as a technician in the honeybee lab here at WSU, and I ended up deciding to stay here as a graduate student because I had this amazing opportunity to work with fungi and bees at the same time, and work on investigating the overlap of ways we can use fungi to help bees. So already, like, we know recently it seems like there's this big boom in popularity of fungi in general, which I think is awesome. It's kind of interesting, like when I walk by the clothes section in Walmart recently, there's always like some fungi imagery on clothing, and it seems like, if you think back to hundreds of years before this, where people kind of just thought fungi were plants or whatever, it seems like all of a sudden there's a lot more popularity. And people appreciate fungi for many different reasons, like just the way that they're beautiful to look at, or the fact that they often have a lot of nutritional value for the ones that will grow to consume. And they often bring people together, like my friends here who I'm foraging with. And there's also been a lot of advances that fungi have been involved with scientifically and medically. So it only makes sense that we should be exploring how fungi can be used to help honeybees, and especially with a lot of the issues we're seeing in commercial beekeeping. So that's kind of how I got here in the Hopkins lab. Here's kind of an overview of kind of what I'm going to be going through today. So I'll give a very brief background. Since you guys are a bee club, I'm going to assume you all are already kind of familiar with the problems we're facing in beekeeping, especially commercial beekeeping right now. Then I'm going to kind of introduce the fungus that's the star of the show, Metarhizium, and how it has already been used in integrated pest management to manage other unwanted pests. And then current and past research already done with Metarhizium to manage Varroa mites. And then lastly, I'll kind of go over what I'm specifically doing in my research project to contribute to this new use of this fungus. So we're already pretty familiar with commercial beekeeping, how we rely on bees as livestock. We're trucking them in and out to these large areas of monoculture. And with that, we depend on them very greatly. I should really update this because they did recently publish something new with the new estimated value of dollars. But in 2000, honeybees were valued at $14.6 billion of worth. So to put it simply, we very much depend on them. And we're currently seeing all these issues year after year, especially in the last few years, where honeybees we're seeing rapid colony loss, where commercial beekeepers coming into the spring after all their colonies have overwintered, they're seeing these massive losses. And my lab actually just recently put out a publication kind of summarizing how it's a result of the 4 Ps: pests, or parasites like Varroa mites, pesticides, pathogens, and then also poor nutrition. So I'm not trying to minimize the problems and say they're all a result of Varroa mites, but we know that mites, parasites are one of the main ones, as well as poor nutrition, and pesticides, and pathogens as well, which are also vectored through these mites. So my project mainly focuses on reducing pathogens and reducing mites in the process. Other way around, reducing mites and thereby reducing pathogens by reducing the number of mites that are there vectoring pathogens to these bees. So we're all familiar with these nasty, nasty guys, the Varroa mites. And originally, these mites were a parasite to Apis cerana. Because Apis cerana had such a long life history with this parasite, it wasn't so devastating to Apis cerana. And it wasn't until like the 1970s that we started or I guess like, yeah, the 1980s, we started seeing these mites spread to Apis mellifera, the bee we keep here in the US. And rapidly, this has led to decades of devastation for Apis mellifera, where we now rapidly need new ways to manage these mites because they've built resistance to many miticides. So with all the problems bees naturally would face in this commercial setting, such as not getting enough nutrition from a vast area of monoculture or losing a lot of foragers when they're transported during the day, and all of these things they would normally face, they're now on top of this having to deal with these deadly parasites that are causing physical damage by damaging their tissues, while also vectoring all these diseases that we're seeing rapid loss from. And we know that there's many different ways to manage Varroa mites, of course. There's mechanical methods such as powder sugar method or destruction of drone brood, but we never really see this done on a commercial level because they are not always reliable or too time consuming. So even if someone's really thorough in clearing out all the drone brood or getting powdered sugar on all those bees, we're never really going to see that implemented on a commercial level just because it's going to be too time consuming. Then we have a lot of risks from synthetic miticides that are building up residues in the comb or having lethal long-term effects in the hive and also damaging hive-derived products like wax and honey. And then there's all these natural chemical miticides, and all of these are going to require proper PPE and planning. So none of these are necessarily reasons why we would never want to use any of these miticides, but just different cons to consider. But the main issue that we're all, I'm assuming, probably pretty familiar with at this point is that mites are building resistance to the synthetic and natural miticides. So as a result, after years and years of repeated use with different miticides, we've seen resistance and we've seen different molecular traits that imply a lot, like specific resistance being built over time. So we need new solutions is the gist of all that. And something that would be very viable to consider would be using something that's a biological control. Currently, there are no biological controls for Varroa mites that are used on a commercial level. There's been exploration of it, but currently nobody really large scale or even really small scale is implementing biological controls to manage Varroa mites. And if you're not familiar, a biological control, it simply just means using one living organism to manage the population of another unwanted organism. So, a simple example that comes to mind would be like using cats to manage for rodent populations, or releasing ladybugs into your garden to manage aphid populations or other small unwanted insects that are going to damage crops. So, but the reason that we would want to consider biological controls for Varroa mites, not only are biological controls easily integrated into sustainable agriculture because they are a natural product, they also have low non-target toxicity levels, meaning usually when you release a biological control, they're not really going to affect other organisms. But most of all, my favorite reason is that usually it's much harder for pests to build resistance for biological controls. So, for example, a rodent isn't going to build resistance over generations from a cat. They might, devise some new kind of strategy to evade a cat or another predator, but we're not going to see this generational change that's based off of like if they have a resistant gene or not to like a certain chemical. So that's kind of the main reason we should really be looking into various options for biological controls for these mites, is it would solve the problem of mites building resistance. And a biological control can be basically any organism. So I mentioned some insects, some mammals like cats and the ladybugs, but birds are implemented as biological controls when farmers will, put perches around their orchard to kill mice. Bacteria are used as biological controls. And of course, the star of our show today, fungi are used as biological controls. And that brings me to, like I said earlier, the star of the show for tonight, Metarhizium. This is a specific genus of fungus that is already used commercially as biological controls for many different insect and arthropod pests. And what's really cool about these fungi is that I didn't know this until I started studying them, but they are widely distributed around the world. And by these fungi, I am talking about Metarhizium specifically, but also many other different types of entomopathogenic fungi, which I'll talk more in a minute. But there are many different types of fungi that parasitize and kill insects, and many of them, including Metarhizium, are found in the soil in basically all climates except Antarctica. So sometimes I think people get kind of freaked out of these fungi because they are zombifying insects, but in reality, these are actually all around us in the soil. Because they don't always need to be parasitizing insects. They can also be saprotrophic, meaning just feeding on dead and decaying matter in the soil. And in that non-infective life cycle, they're also beneficial fungi because they are contributing to nutrient cycling and breaking things down. So yeah. And yeah, here's what that would look like. So fungi grow as mycelium, which is sort of the vegetative form of the fungus. If you're not familiar with mycelium, it's the white threads you might see if you pick up a mushroom in the woods or if you roll over a log. And this is the main part of the fungal organism. But then eventually, mycelium in most fungal species, this one included, produce spores. In these types of fungi, the spores are often referred to as conidia, so that's why it says conidiation here, but basically spore production. And then these spores, which are the infective propagules or the dispersal propagules for other fungi that aren't infective, are then spread through the air, or in this case, in the air or the soil, and then germinate, form more mycelium, more vegetative growth, and then continued sporulation. So this is what that non-infective life cycle would look like for Metarhizium. But I don't really care about that. I don't care about the non-infective life cycle because what we want is the infective life cycle. And here's what that would look like. So that spore that's produced, it can either end up in the soil and continue that vegetative growth that we just saw, or if it happens to adhere to an insect's cuticle or exoskeleton, it will start doing something completely different. Basically, the spore adheres to the cuticle, and it'll send out what's known as the germination tube, often called the germ tube. And this is like basically the first strand of mycelium that's produced. But these fungi are so specialized that this germ tube will start secreting very specialized enzymes that have evolved over millions of years to be able to specifically break down the waxy, lipid-rich layer of the insect's exoskeleton. So very specialized, and it uses these enzymes in a combination with turgor pressure. So it's pumping fluids into this structure and using that to break into the insect's exoskeleton. So very important to note, the insects are not eating these spores. This is not something that is infecting the insect through digestion. It's purely breaking through from the outside from its cuticle. So very remarkable. And once inside the cuticle, the fungus will start proliferating as this vegetative mycelial growth inside the insect's hemocell or body cavity. So yeah, it'll slowly like feed on the most non-vital organs first, and then eventually as it grows throughout the whole insect's body, it will then feed on the most vital organs, killing the insect, which is also another really interesting feature that has developed, because if you think about it, if the fungus killed that insect right away, it would immediately be competing with all kinds of other microbes that are trying to grow on that decaying body. So very, very specialized. And then once the fungus has proliferated, it will produce more spores or conidia on the outside of the insect. So that's when we get this fuzzy growth here. Yeah, and the fungus is, of course, doing this to exploit nutrition. However, you can also think about how this is probably how these fungi spread all across the world, because if you're like this sad little grasshopper who gets infected by metarhizium or whatever, you're probably going to hop a great distance before you're dead after several days, because this whole process is taking, depending on the size of the insect, between 3 and 12 days. So the insect's going to travel, and it's a dispersal method as well. But what's really cool is how this fungus can flip flop between these two life cycles. It can be non-infective and just be saprotrophic in the soil, or if given the opportunity, it can infect and kill these insects. So super cool. And yeah, here's just another image kind of depicting what I just said. So spores get dispersed and adhere, and then they break in, grow inside with this mycelium, and then produce more spore-producing structures on the exterior of the insect. And, I'm going to talk about metarhizium, this specific green, dusty fungus, for a while. So I just want to emphasize there are so many different types of fungi that also have similar life cycles. And you can find them outdoors. You can find them basically anywhere that is like a really humid, moist climate. And some of them are really cool. So just before I talk about metarhizium for so long, I guess I already kind of summarized metarhizium, but before I talk about that with the bees, I just want to highlight, here's something you can go find in Florida that I just found a couple weeks ago. But this is the fungus that The Last of Us was inspired by. If anybody's seen that show, people always ask me about it because of this, but these are ants. This fungus is specific to this species of ant, and it's very unique in that it will manipulate the ant's behavior by making it climb very, very high. Well, for us, eye level, or for me, eye level. I'm 5'2", but it'll make the ant climb up high on some kind of overhanging vegetation. And we don't entirely know how it does this. We know that the mycelium is infecting the nervous system of the ant and somehow manipulating it to do this. But the ants will climb up and they'll do what's called the death grip with their mandibles, where they grip down on this blade of vegetation that they're on, and then they die. And then this fungal fruiting body structure emerges from the back of the ant's head, and this is covered in spores. So the fungus is manipulating the ant to get into the optimum location for more spores to then be dispersed down on other ants that are trailing underneath these plants all the time. So really awesome and unique how different these fungi can be, and how specialist they are. And yeah, so if you're ever in the Hoh rainforest, or I'm not sure what the equivalent down in Oregon, what other things are around you guys, but if you go to any super humid, moist area, look specifically if there's a creek flowing and a bunch of vegetation above it that's kind of creating a little humid chamber, that can be good locations to look. So feel free to email me pictures of whatever you find, because I love finding stuff like this. But back to metarhizium. Metarhizium, like I mentioned, it is a genus, so there's many different species of metarhizium. But it's known for producing these green, dusty spores on the surface of insects. And depending on the species of metarhizium, it can be either very generalist, where it will infect many different kinds of insect pests, or it can be very specialist, where it's only infecting specific species, kind of like that Ophiocordyceps only infecting the ant. So depending on the species, can be very different in the host range. And surprisingly, we've actually known about this fungus for quite a while. So this is Eli Metchnikoff. He is kind of known for discovering a lot of early microbiology kind of stuff. I really only know about him because of metarhizium, but he discovered other microbiology related things. But he first observed metarhizium growing on a wheat grain beetle in 1879. And you can kind of think about how a wheat grain beetle probably would have been more common of a pest then, right? People are storing grain in their house probably, and this would have been maybe a pest people dealt with. And after he observed that, only 5 years later, he was using this to kill the wheat grain beetle. So he had cultured the fungus from a dead beetle and propagated it, and then they were using it with reportedly very high mortality rates, 80%. Well, up to 80%, it says here, but still useful for the situation that it's killing the pest. Now, flash forward to today, there are a variety of products using Metarhizium fungus as the main active ingredient. So here's a popular example is Met 52. And this would be an example of a dry application of this fungus as a pesticide, where people are purchasing a bag of spores in a like carrier powder and distributing that. But it can also be distributed in an aqueous solution. And most of the time, these fungal products are being distributed either into the soil to boost sort of like the natural population that maybe would already be present in the soil, or they're applied directly onto plant crops where, you know, an insect pest might come chew on the plant and come in contact with the spores and then become infected. And of course, the way that these products are applied is going to like completely different efficacy depending on the conditions of the application or the way in which they're applied. So that's when different carrier powders, carrier solutions, or different application methods kind of come into play. But also, it's going to be limited by different things that the fungus is going to prefer, right? So we would want to have optimum conditions for fungal growth in order to see optimum performance of the fungus. And looking kind of closer at those spores I mentioned, it's always the spores that are getting distributed because that's what carries out that infective life cycle. So there have been studies where people grow a whole bunch of Metarhizium mycelium and put that on insects, and it will do nothing. So importantly, it's these spores that are the infective propagules that do the infecting and that we're using in products like this. So I'm really lucky that we here at WSU, we have a electron microscopy lab that you can like pay for a membership to. So I was able to get these images of Metarhizium spores. So this is, you know, you can't really see the spores quite yet in this image, but if we were to look at the agar plate and it's like a green agar plate, like I showed in a previous slide, that's just dust that is spores. And so this is that from afar. You know, we're still pretty close, but if you get closer, you'll see these spores. And Metarhizium specifically is known for producing spores in this chain formation. So that's what's used to identify it with microscopy. And yeah, it was awesome to be able to get these images because I ended up learning a lot. So for example, these grooved textures on the spore are known as rodlet bundles, and they aid in making the spore very hydrophobic, which helps with adhering to insect cuticles, which are also hydrophobic. So hydrophobic surfaces want to adhere to each other. And this is another awesome feature that these fungi have for making them so great at infecting and killing insects, is they have these bundles of proteins giving them this extra hydrophobic texture. And there's actually been some papers where they somehow were able to like delete the gene that makes these bundles, and they were able to propagate these like really smooth surfaced spores, and they really sucked at infecting insects. So just little things like this play a huge role in the performance of these fungi. So with all that being said, what in the world would be stopping us from just dumping a whole bunch of spores in the beehive and letting it kill the mites? We know, I'm going to talk about it a little more later, but we know that these fungi don't really infect bees, luckily, because the bees are able to clean it off of themselves. But what's limiting its effectiveness against the mites? It's temperature. So because these fungi naturally are usually doing this thing outside of a beehive, in the soil, you know, under plants, wherever they're coming in contact with insects, usually, they're not used to the high temperatures found inside the beehive. So there have been other papers where other labs that have investigated using these fungi in beehives, and what they found was the fungi could kill some mites, but not enough to be as effective as the commercially available miticides. So Yeah, this has been kind of a major limiting feature in developing these fungi for Varroa mite management, is the fact that we know they are not very thermally tolerant, because naturally they don't want to germinate in these high heat environments. And like I mentioned, germination is that key step. Like if we could grow mycelium at high temperature, it wouldn't matter. We need germination specifically, because that's how that infection starts. So in 2021, some researchers here in my lab, this predated me. I joined the project after this paper was already out, but two of our postdoctoral staff were able to select a strain of Metarhizium that was able to germinate in these high heat environments, and they performed repeated generational selection using this method. So basically, they grew spores on agar, and then they applied one type of stress to each generation, either oxidative stress by pouring hydrogen peroxide onto the agar plate, or they use nutritional stress by growing the fungus on a nutrient deficient agar media. And the reason like we would want to apply stress to these fungi is that, and you know, I can't get too into explaining it because it gets really complicated, but basically these are asexually reproducing fungi, meaning each generation we select, there's not going to be as much differentiation as there would be with a sexually reproducing fungus. So applying stress like this aids in mutation, which is a faster way to get variable results when doing strain selection with this type of fungus. So they'd apply stress, continue propagation, and then mix up the spores in a new solution and incubate them again on more agar at an increased level of temperature. And they continue this over and over with more spores being produced in each generation. And then from there, they basically just inverted an agar plate with these spores on it onto a beehive, and then monitored the mortality of the mites by using sticky cards in the bottom of the hive. And they were able to reculture Metarhizium from mites that had been successfully infected. So they collected dead mites from the sticky cards at the bottom of the hive, surface sterilized them, and placed them on agar media here. So each of these spots in this image is going to be where that green dusty fungus is growing from a plated mite. And like if you see down here, oh, there's one spot missing. Well, that's probably maybe a dead mite that was plated that was not infected with Metarhizium, so it did not exhibit any fungal growth. And from there, they were able to continue propagating the fungus from these successfully infected mites, and they basically witnessed like improved performance of the fungus after passaging it through the mites like this. So they did another field trial and they were able to see, okay, now it's killing more mites. And here's a very good visual for the results of this. So I mentioned the spores are going to produce a germ tube when they germinate. That's what we're looking at here. So these little tubes emerging from the spores are the germ tubes. And you can see in this top row, there's not very prominent germ tubes action going on here compared to the lower row. And that's because this is the fungus without any selection, versus down here is the fungus that was selected over multiple generations to be heat tolerant. So this is just a very distinct way to kind of see, okay, yeah, the performance is improved at higher temperatures because these germ tubes, they're ready to go and infect something. They're reaching out. So when I came onto this project here, we were cultivating the fungus on brown rice in order to then put it in the beehive. So this is what a lot of that work looked like that I was doing early on, where we would autoclave rice to sterilize it in these unicorn bags, which are traditionally used for growing gourmet fungi. They'll like fruit it out of the sides. And then we'd have agar plates of the fungus that we put in the rice and allow it to grow all along the whole rice. And yeah, but because we went through so much rice, me, had to make a lot of trips to Costco getting our very specific fungicide-free rice, organic. And then we would weigh out 400 grams of this rice, which it's no longer just rice, right? It's rice that is green and dusty with these spores. And we would put 400 grams of that sporulated rice into one of these paper bags. And then you can see we kind of have a spacer on the top of the hive here to give it an extra couple inches. And my colleague there is cutting a little hole in the bag to basically initiate that disturbance so that the bees will want to interact with it and want to clear that debris out of their space, and in the process hopefully get all those spores all over themselves and each other, and then be cleaning it off of each other, and in the process kind of distributing it on all the mites. And yeah, so when we did this field trial, we monitored the mite population by putting these sticky cards in, and we just make these sticky cards. It makes it sound like it's this fancy product, but really we're buying a bucket of Vaseline, and that's another interesting purchase other than the rice. Is we're getting buckets of Vaseline and coating them onto these paper cards, and then putting a screen over top and sticking them in the entrance of the hive. And you can see here in this image, there's like a lot of dark spots, and that's actually some of the old rice grains that fell to the bottom after they had already been disturbed and kind of dried up and were able to fall down there. So the bees are distributing that rice, and then we're able to draw these mite cards out and count the number of mites that are dying, or at least that are falling to the bottom of the hive. But there should be a difference between control groups and treatment groups, right? If even though there will be a natural amount of mite fall and mite death, we should notice a distinct difference if the fungus is killing more mites. Oh, and there's a mite. So we did that field trial for a summer. That was a couple years ago. And while we were seeing some increased mite fall, we did not quite get enough data to really publish anything. And, it sounds like a real bummer, but if you're in the world of research and everything, well, this kind of happens a lot. And sometimes when you're trying to take something that's supposed to work in a specific way in field conditions and it doesn't quite go right, sometimes you kind of just need to take a step back into the lab and kind of look at things in vitro a little more. So this is what we did after that experiment. Another thing too that we learned later on is that apparently, as far as EPA approval for a product, you would never be allowed to have a human food ingredient in it. So even if the rice thing worked, we would have to eventually try something else if we're trying to implement this as a product on a commercial level, which is the goal here. Because rice, that cannot be part of a pesticide product if it has a human food ingredient. So anyway, we looked back, took this into the lab, and we basically did this bioassay where it was very tedious. I'll show a video in a second, but the short of it is we incubated mites on bee pupa in these centrifuge tubes in the lab, and some of the mites we treated with a spore solution, and essentially we're replicating the conditions in the hive, but doing it in the lab. And the reason we need a bee pupa is because these mites would just die of starvation, right? If we collected a bunch of mites and treated them and had them in a little dish, they would just die. So we had this tedious process involved with pulling out pupa to allow the mites to feed on them, so we could actually observe if the fungus was killing them. So just setting it up, here's kind of what this looked like. It took a lot of hands it still took all day, even though we had 6 people helping us, but essentially we're picking through these brood frames and removing both last instar bee pupa and mites. And if you're ever needing to pick up mites for any reason, a camel hair paintbrush is a very good way to do that. We can kind of just twirl them, and their little legs are and they just kind of are there on the paintbrush. That's how we set it up. And then here, this is me making a spore solution with some of our Metarhizium colonized rice. So I'm basically swishing the rice around in a carrier solution, and then filtering it to get this solution with spores in it. And then my friend here is taking the mites putting them on a filter paper, and we're pouring this solution over them, and then picking them up with our handy dandy little paintbrush and putting back on the pupa in the tube. And I mentioned it was in a control solution. We were using Tween 80, which is basically a surfactant. It prevents the spores from clumping up, and it's used in a lot of different pesticide products. And I mean, it's water with like 0.05% Tween 80. So that was the carrier solution the spores were in. And then the control group, therefore, was just dipped in the, or they just had the control solution poured over them. So that we could see if, it wasn't just the carrier solution killing them. And we monitored for mortality daily. We checked each little tube with the pupa and made sure each mite was alive. And if it was dead, pulled it out, surface sterilized it, and put it on another agar plate. And therefore, the idea with this, just like before, is that if there's any fungi growing inside this dead mite, it should grow out on the plate. And that's part of the deal with surface sterilizing is, there could be fungi naturally occurring externally on the mite. And yeah, if that's confusing to anyone, just know, there's fungi in the air all the time. So if we were to touch any of these mites or anything from outside and put it on the agar plate, it would grow like mold or whatever's in the air. So that's why we're sterilizing the rice and surface sterilizing any insects that we're trying to grow fungus out of, because otherwise there's going to be natural fungal contaminants that would just grow on the plate. So anyway, we surface sterilize it to see if there's any fungi inside that will then grow out. And you're able to see here, we did get some fungal growth here. But what's more important, even if we don't get fungal growth occurring, there was a significant difference between mortality. And, I could get into more detail on it, but basically, when insects are killed by these fungi, we might not necessarily observe spores on the outside of the insect if it's not ideal conditions for sporulation. But if we're still seeing a distinct difference in mortality, that's still useful information where we're able to quantify how effective the fungus is. So for the ones that did exhibit fungal growth, we see this nice green growth in the middle. And yeah, so that's what we would expect from Metarhizium. And here's that mortality data that we care the most about, or I do anyway. But basically, you can see by day 5, we had over 50% higher mortality in the treatment group than in the control group. And this was statistically significant. So that was pretty satisfying to see that. So I kind of touched on it earlier, but everyone always wants to know, well, how do you know this fungus isn't just going to kill all the bees? Because that's like the exact opposite of what we would want. And I kind of mentioned it. Bees, they're so hygienic. They're cleaning themselves. And if you think about how the bee's body is kind of made for collecting pollen spores, these pollen spores are held away from its cuticle. So you can kind of think of like any spores that are going to be on this fuzzy adult bee are kind of away from the exoskeleton, and they're away in such a way where the bee is then able to clean these pollen spores off and like pack them up. So in a similar way, any fungal spores that land on the bee, they're probably getting cleaned off. And like I mentioned, there have been a few other labs, a couple through the USDA that did do research for a couple different years on these fungi in beehives. And they took a lot of data on like the strength of the hives, and they had dead bee traps to observe, to quantify how many bees were dying each day. And basically, we're very, very confident at this point with previous research that these fungi do not infect and kill adult bees, or at least not in any way that's really significant or measurable. There have been some papers that specifically take brood frames, cut a little slit in one of the cells, and inoculate a spore solution. And they did observe low mortality rates with those larvae. However, that's a situation where the spores were directly inoculated, and it was under 10% mortality. So that, even if that was occurring in a situation where we were treating this in field conditions in a hive, that amount of mortality could be acceptable if it's also wiping out all the mites in that brood cycle. So maybe Obviously we want to continue doing research on how this is affecting bees as we develop a product like this. But so far, it seems like if there are some amount of larval mortalities, it could be an acceptable trade-off as long as it's effectively killing these mites. And yeah, so bee hair, hygienic behavior. Also, another interesting thing I just want to add in there without going off on a tangent too much, but in that study where they were directly inoculating larval cells with metarhizium, they also took metarhizium and put it on Varroa mites and then inoculated those larvae with those fungal inoculated Varroa mites. And what was really interesting, at least in that study, we'll need to repeat this more to know more or to, be certain, but it's been observed that larvae infected with Varroa mites that are infected by metarhizium had higher immune responses, like based on the expression of their immune priming genes, than larvae infected by Varroa mites. So it's been known for a while that Varroa mites suppress bee immune systems. And essentially what they saw in that study was that when Varroa mites infected with metarhizium were in the bee brood, that immune gene suppression was canceled out. So, like, I'll talk about it a little more, but in my future plans with this research, we're going to be looking at that more. But basically, like, treating metarhizium in a hive could cancel out the immune suppression caused by Varroa mites, which could be really, really important to know as we're dealing with, mites transferring pathogens and viruses and diseases. That could be really useful if the bees coming in contact with these mites infected with metarhizium are then more robust against these different stressors. So next steps, I'm going to be doing a field trial this spring and summer with these fungi, and also the next year after that as well, the 2027 field season. But we're going to be working with figuring out the best modes of application for these fungi. So I mentioned we had been putting rice in the colony, and you can kind of think of that as a very passive form of application because we're relying on the bees to, use up their energy, removing the debris, and in the process, cleaning it on themselves. We're kind of just putting something in the hive and allowing the bees to put it on themselves. It's a very passive mode of application. Kind of like you can think of putting in a rag that's soaked with oxalic acid or whatever. But there are a lot more active or direct modes of application that we could explore as well that might be more effective. So you can compare the rag option to like vaporizing with oxalic acid or doing the dribble where you're actively kind of forcing contact. So in a similar way to there being these different options with something like oxalic acid, we could explore various options with different amounts of passiveness and directiveness than with these fungi. So something I really want to try that I think would be cool would be using some sort of aerosol sprayer or some kind of spray option to just spray like a dry spray of spores throughout the hive, kind of in a similar way to vaporizing oxalic. Because whatever mode of application works the most effectively at killing mites, it also needs to be very efficient if it's going to be integrated on a commercial level. So that's kind of something that we got to keep in mind the whole time is whatever way is the best to apply this, it also has to be fast because no commercial beekeeper is going to do something that takes more than a couple minutes, or even that is like a long time to spend per colony when you've got thousands of colonies, right? So if it's something that somebody could go around and just like stick in the entrance of the hive and like it sprays, you go to the next one, that would be ideal. So with that being said, we talked a lot about metarhizium. There's all these different genera of fungi, and my research specifically, I kind of touched on it. I'm kind of doing a lot with this project in general, and there's a lot of different kind of projects within the project, but I'm specifically comparing thermal tolerance selection over multiple generations with metarhizium, Bovaria, and Hirsitella. So these 3 different genuses of fungi are the fungi I work with. And basically the idea behind that is Bovaria and Hirsitella, these are also widely used as natural pesticide products. Opening it up to more than just one genus of fungi means more potential to find something that works really, really effectively at high temperatures. So here's kind of what I've been doing the last like 6 months to contribute to this is I start with an initial screening. So I grow these fungi on agar and I incubate them at beehive temperature. So starting at 32 degrees. And you can see, so like here's a photo on the right from my initial screening. This top row of plates was grown at 27C. Then it was like 29C, 31C, and then like 33C. So you can, yeah, degrees Celsius. So you can see, the opposite. These ones that grew the best are the ones that were incubated at 27C, and so on. So you can see this top row here had very, very little growth after a few weeks, because these fungi are not naturally inclined to grow at 34 degrees Celsius or whatever. So performing this initial screening is kind of just a way to see, okay, here's like the temperature limit of where these fungi want to grow. Yeah. And again, that's just looking at the vegetative growth. So what I then do is basically I isolate an individual spore from the highest performing fungi that grew vegetatively. And but I want to isolate a spore that's successful at germinating. So essentially I'm making spore solutions and putting them on more agar plates. Surprise, surprise. It always goes back to an agar plate, right? But basically incubating them on agar for 6 to 10 hours. And then I come back and observe the germination. So here's more of these germ tubes that I would use to quantify, okay, here's this one's germinated, this one's germinated, this one's germinated, this one's not. And I would count 100 spores to get like a percentage of the germination rate. And from here, I isolate an individual spore. And this is kind of what separates these methods I'm doing and makes them different from what the previously published paper did. Was they selected a strain based on phenotype. They had observed the fungus was growing at higher temperatures, but there was no singular genome that was being worked with. So my goal in selecting these strains is that these can be strains that are distinguished as unique genomes that have specific molecular markers that would distinguish them from other isolates of the same species of fungus. So, and with that, I'm able to have more uniformity among selection. If one spore exhibits some specific gene that allows it to be more likely to germinate in the high temperature, then its progeny are more likely to have that same trait, as opposed to just taking a whole bunch of spores, right? That would be multiple genomes. So I take those successfully germinated spores individually on new agar, grow them out again. Here's a picture of some mycelium that was taken under our microscope camera here. So I allow this vegetative growth to occur, and then basically continue this process again, where I allowed this mycelium to produce more spores, and then I observe that germination rate once more. And it's important that at least every few generations, I need to passage these fungi through some sort of insect or arthropod host. Because what can happen if you're growing these fungi over and over and over on agar plates, the fungi are going to get adapted to just growing on agar plates. They're going to become kind of wimpy and kind of lose their traits that allow them to be virulent and effective at killing things. So because of that, I had to learn how to rear wax moth larvae in the lab. And usually when I mention this to beekeepers, they're like, well, hang on, what? You were rearing wax moth larvae? I'm like, listen, they're very useful in the lab because they're easy to rear in like a box with just some wax or whatever. And they're really easy to apply spores on and just observe their death and mortality. And because they melanize, they turn dark when they become infected, I can observe this infection very, very easily versus having to like poke and prod a mite to see if it's still alive. So that's kind of an important thing, is passaging it through a host. Otherwise, these fungi become less and less effective. So kind of here's a summary of what everything I just covered. So initial screening, starting that first generation, isolating individual spores after measuring the germination rate of those replicates, and basically picking the ones that have the highest germination rate and isolating individual spores from there. And then every so often confirming virulence, but ultimately continuing this back and forth cycle of selecting spores, allowing vegetative growth, and selecting spores again, all based on germination at 32 degrees Celsius and above. And here's where I'm at with that. Currently working on the strain selections, like for the next couple months and for the past several months. And then, like I said, field trials are going to be occurring this spring and summer, where I'm going to be directly comparing the effectiveness of the fungi I've selected for thermal tolerance compared to the fungi that have not been selected. So there will be a treatment group with Beauveria that has not been selected, and a treatment group with Metarhizium that has not been selected. And then there will be treatment groups with the ones that have been. So not only kind of continuing to see, okay, well, how can we make this fungus effective, but also seeing, okay, well, are these methods useful? Because again, my interest is with fungi, and if these strain selection methods work very well at resulting in thermally tolerant strains, you could use them to select for other relevant stressors. So, like, for example, UV radiation is just from the sun is something that will kill these spores over time if you're talking about, applying these spores on a plant crop. So perhaps in the future, people can use similar methods where instead of incubating at a high temperature, maybe they incubate under a UV light or some form of sunlight or something like that, and then select germination rates from there and select single spores from there. So I hope that these methods I'm developing can be broadly applicable for strain selection in general, for developing fungi useful for different situations. So after these field trials, we're going to be continuing to confirm the application methods, and then there will be molecular work done along the way to, like I said, figure out how we can distinguish this strain from the previous fungus it was selected from. So, for example, that could look like something like, well, this one heat shock protein gene is massively upregulated in the resulting strain, or something like that. There should be some kind of molecular marker that can be used to then distinguish it. And I didn't really show it in here or mention it fully, but there will be samples of bees taken that all along the way throughout the field trials, right after exposure, before exposure, and then like a week after exposure from the fungus, just to both confirm that the fungus is safe for bees, but also further investigate the effects of the fungus on the bee immune system. Because if we continue to see that Metarhizium perhaps upregulates all these immune priming genes in the worker bees, maybe, and of course that could maybe be because on a molecular or on a very small level, their bodies are like fending off this pathogen, and it's like priming their immune system for anything else that they're going to come in contact with. That could be really awesome and beneficial to know. So could have secondary beneficial effects other than killing mites. It might help bees be more robust against pathogens. And then ultimately, the goal is to have EPA approval so that beekeepers can then use this as a registered treatment in their hives. So to conclude, we know that there's all these problems with honeybees. There's different factors, but for me, it's the most relevant one is the mites. We want to get rid of these mites, and we need new methods. A biocontrol could be a great option because it would likely be more sustainable long term, because organisms are less likely to build resistance to biological controls. And Metarhizium and other fungi show promise to be this first biological control. So, but in order to do that, we got to develop thermally tolerant strains and figure out a couple more things, like what's the best way to apply. But we're going to be continuing to investigate all of these things, and hopefully you'll hear more from me in the future of something positive. But I hope that this has been a good way to kind of show how fungi can be used to help our bees. And yeah, I'd like to thank everyone in my lab who, they're not here, but this is a big group project that has involved a lot of different people. This is my thesis project, but like all projects in the bee lab and any situation with bees, usually it's a team effort, and you need a lot of hands on deck. So they've all helped me a lot through this. And then thank you to the Washington State Beekeepers Association for funding my project 2 years in a row. So shout out to them. And with that, I'll take any questions or comments or anything. And if you like, you can email me questions. And if you do find any entomopathogenic fungi out there, feel free to email me pictures of those as well, because I will never say no to a good fungus picture. So yeah, thanks you guys for having me.
Yay. We can. Okay. There we go.
So I've got a question for you, Molly.
Yeah.
So you talked about the bees grooming themselves and therefore getting rid of the spores and so forth. And I know you mentioned about doing further studies perhaps to make sure that it is safe for the bees. But do you think there's any possibility of, once the fungus has run out of its host, i.e. the mites, that it might evolve to start attacking the bees? How specific are they, in other words?
That's a really good question. Personally, I do not have, based on like different research I've seen, I do not feel very concerned about that. But another thing, you mentioned host specificity, and I kind of, well, I guess I mentioned it with the other paper that we published, that my lab published a few years ago. But like, I intend to continue selecting these strains from Varroa Mite cadavers that have successfully been infected by the fungus. And in doing that, I hope to develop host specificity with this strain towards mites specifically. So I think that if that is possible and I'm able to do that over multiple generations, we should see less and less risk of this fungus towards bees, just because mites are so different, like structurally and biologically. But yeah, like I said, we want to continue to monitor that. And yeah, it's also another thing to consider too is like you mentioned, when it runs out of its host or whatever, these fungi can also just like be dormant. It's not like a mammal where it like needs to keep consuming to, in order to live. And, so basically if there was no suitable host present, these fungi could just like be there and not really do anything until there is a host present. They're not necessarily growing out like needing a host, because they can continue that saprotrophic life cycle. So, but yeah, like absolutely with any type of pesticide or biological control, we want to keep monitoring that. So, I tend to do everything I can to make sure that this is totally safe for bees as we develop the strain.
Thank you.
I have a question, Molly. My question is, have you looked at all in your study about how it could be, I saw on one of your slides it noted that it was minimally like toxic to humans, or I guess it didn't say toxic, but something like that. Is there anything that would be problematic for consuming the honey or using the comb that has to do with this particular fungi?
That's a really good question. And short answer is that probably more research will need to be done, like on hive products that have been treated with the fungus, in order to truly know that. But yeah, I think I kind of skipped over it. But yeah, on one of my slides, I said these fungi have minimal risk to humans and mammals. And when I say that, like kind of the minimal risk is just that it's not good to breathe spores. So if we were applying this in like a dry form, we would want to either make sure it's being applied in a way where it's not, dusting out on the applicator, or maybe the applicator will have to wear a respirator, something like that. But, it's never good for people to breathe spores. So that's kind of what I meant by that little thing I put in there. But yeah, as far as hive products, we'll have to do more investigating with that. But kind of one of my thought processes, just kind of just speaking on my thoughts, not no data really to back this up at all, but there's a lot of spores present in the environment, and therefore, like, they're always going to have spores from outside on their bodies. And of course, that would be completely different than if we were directly dumping a whole bunch of spores right on some honeycomb. Obviously, it's going to have more exposure to fungi, but there's likely already a lot of fungal spores in comb and in honey. Because we know there's pollen spores mixed in with those things too. But yeah, like, so depending on what we find, there very well could be like some limitation with the usage, like maybe you don't apply it in your honey supers or something like that. So more research will need to be done.